way back. And Nick grew up here in town, and you know, he graduated from here. He went to Kalamazoo College, but then majored in all things philosophy.
Yeah,
he got degrees in philosophy, but made his way to epidemiology and public health. And Mike Benke, who's out of town today, was his PhD thesis advisor. And then, you know, he entered the academic track and started off at Case Western Reserve in Cleveland, and then started migrating to the West Coast. And he's got lots of different positions here at the West Coast now. You know, with Craig Benter at the J. Craig Benter Institute, where he's a professor and director and oversees the next-gen sequencing kind of activities there, which is really saying something given what they're doing with Illumina. He's at T-Gen, where he has an appointment there as a professor. You know, he's been involved with the Human Longevity Institute. Interesting story there. He's been at Scripps. You know, he was with, I think many of you know, he was with Eric Topol at the Scripps Translational Research Institute before he went to work with Greg. He's adjunct in several departments, including psychiatry at the University of California, San Diego. And he's a great friend. And he's a visionary in terms of how we might develop, in what he will be talking about today, are a new class of clinical trials. They're adaptive, they're statistically reasonable, and they're personalized. And I think I pretty much summarized the title of your talk, and it's just wonderful to have you back.
Great.
Without even looking. Okay, good. Wonderful to have you back.
Okay. Well, it's a real pleasure to be back. And as Brian kind of alluded to, and as my father and Pat know all too well, sometimes I have like lots of material in my talk. So if I go on for a long time, Pat, whip out the hook and just get me off the stage, as you've done many times in the past. So, so here are the topics that I want to touch on today, and I have maybe 5, 6 slides for each one. First, talk about how one goes about identifying variants that influence idiopathic disease, and how one might be able to design studies around treating people based on the insights obtained from identifying those genetic variants. Then I'll talk more specifically about the design of single subject clinical trials. This has been a growing area of interest of mine. And in particular, talk about not just the motivation, but the design of those trials, and have examples of trials we've actually pursued. Then talk about cancer genomic trials and this concept of vetting algorithms versus vetting compounds. This is an emerging theme in oncology, but it comes with it all sorts of repercussions for the design and implementation of trials. I'll also touch a little bit on my exchanges with the FDA about the design and implementation of these trials. Then talk about actually monitoring people for early signs of disease and the need to pay attention to personal thresholds versus population thresholds. This concept is very intuitive, but it's not often exploited in kind of preventive medicine studies. And then I'll just close with a comment on how epidemiology shapes personalized medicine, and personalized medicine can shape an epidemiological studies. So before that, some financial disclosures. As Brian knows, I also have my hands in a few companies. The other thing I'd like to point out is I'm actually on the external advisory board for MISHAR here. So I come back every now and then and review the programs in translational science here. Really flattered to have been asked to be on that board. Okay, so what is it about precision, individualized, or personalized medicine that makes it so interesting in this day and age? Well, not so long ago, there was an editorial in Nature Biotechnology asking whether personalized medicine was finally arriving. And there was a lot of buzz that surrounded the introduction of personalized medicine into the community. So much so that textbooks were written. Like here, a former chairman of mine, Hunt Willard, wrote a textbook and talked about the need for med students to be aware of emerging themes in personalized medicine. Ironically, 4 years later, Nature Biotechnology published another editorial, What Happened to Personalized Medicine? So one has to, what was the deal? What I believed happened is there simply wasn't enough attention paid to how one actually vets personalized medicine to show that it actually leads to better outcomes than traditional approaches to practicing medicine. So it's in this context. I agree with that 100%. Just getting buy-in from the funding agencies is another question. So at any rate, it's in this context that I want to go about giving the rest of my presentation. We'll first talk about identifying variants that cause idiopathic diseases and the filtering strategies that are used to identify the variants that one might build some insight around to craft an intervention for someone with such a disease. So these are pictures of children mostly that have had idiopathic diseases. And for those of you that don't know what an idiopathic disease is, an idiopathic disease is simply a disease that defies easy diagnosis. Every hospital system on the planet has some number of patients that come into them that present some kind of mystery to the physician. And they typically get passed around from clinic to clinic, physician to physician, to see if some insight can be obtained as to what is actually bothering these people. And here, I'm not talking about diseases like fibromyalgia and other ones that have kind of vague neurological symptoms. I'm talking about severe, debilitating conditions of the type that, say, Nicholas Volcker had 100 or so surgeries before he was 5 years old, with a serious gastrointestinal problem, and no one knew actually what was going on. What was ultimately done to these people in particular is their genomes were sequenced. The idea being that if they could identify a variant that showed some kind of defect that was consistent with the molecular physiologic problem that could explain their condition, then maybe they could craft an intervention around that insight. So all these people have had their genome sequenced. With the exception of one, there have been treatments fabricated around the identification of the pathogenic variant. I've had the pleasure of working with Lily Grossman here, and I'll explain a little bit about my experience sequencing her genome with my colleagues and coming up with some kind of therapy that showed some benefit to her. So the real question is, how does one identify the variants that are responsible for these idiopathic conditions? These are true N-of-1 studies. In fact, the assumption is that the variants responsible for these people's diseases must be unique to the genomes that these people have. So think of de novo variations that these people possess that cause disease. Now, why is this the case? Well, if in fact other people on the planet have a similar condition, then this person's disease would not be idiopathic. It would be shared with other people. So if one makes the assumption that the variant responsible for the disease that these people have is unique to the genome of the individuals, then you should not see those variants in any other genome. So as part of a filtering strategy or a strategy for identifying the variants responsible for the disease, one could sequence the patient's genome, look at variations in those genomes, compare them with other genomes, say reference genomes, and only those sites that are present, or variants that are present in the patient's genome, are candidates for being the pathogenic variant. Now, there's a little bit more to it than that, because each of our genomes has anywhere between 5 to 10 to 15,000 unique, probably de novo variants. So the question is, how could you filter them even further to maybe stumble on the offending or pathogenic variant? Well, the idea is to leverage annotations of one sort or another, kind of annotations about what the nucleotide substitution might actually do. So what one could do is throw out all the variants that aren't novel or unique to the patient's genome, and then ask questions about the remaining variants and what they might do in impacting whatever functional elements they might reside in. So you could ask whether or not, say, a coding variant that is unique to the patient's genome actually damages the encoded protein of the gene it sits in to a degree that might explain the disease. Or if the variant sits in a microRNA binding site, is it likely to disrupt microRNA binding to the point that it brings down the functioning of a particular gene or system? So what one can do is simply filter down all the variants in the individual genome to hopefully arrive at a pathogenic variant. Now, this is not trivial, but it has been done, and done with some success, as I had alluded to earlier. So I'll just talk about the strategy for making claims about the functional significance of these nucleotide substitutions that populate the genomes of people with idiopathic disease, and then talk a little bit about how one decides whether, in fact, they're novel and what kind of reference databases are needed to make the claim that variants are novel. So here's just a cartoon of a gene that I stole from a textbook that was published a little while ago. And it just has all the elements in and around genes that, if disrupted, might cause the gene to dysfunction. And with current computational tools, it's possible to predict whether or not nucleotide substitution in these various functional elements likely damage those elements. Now, this is not an exact science, but it's better than a flip of the coin. And you might ask the question, well, why does one have to rely on computational tools? Well, it goes back to what I said before. There's probably too many variants to subject to kind of functional assays to make claims about whether or not those variants actually do upset molecular physiology to the point of leading to some kind of pathology. So suffice it to say, the community as a whole has been developing tools to annotate variations that could be used in this filtering process. My lab over the years has kind of contributed to this literature, but many, many other groups have engaged in this as well and had some success in being able to differentiate variants that are likely to damage the functioning of a gene from those that aren't. Now, once one has a variants that look as though they might be of functional significance, now you also have to couple that with which sets of variants are novel to a particular individual's genome. So I want to take a step back and ask, what kind of diversity do humans show at a functional level in their genome on a global scale? So some years ago, in collaboration with Complete Genomics, which was the only whole genome sequencing player in the community, we sequenced the genomes from individuals from different populations around the world. We took all the variants and subjected them to those computational tools that I described to assess the functional diversity of these variants on a global scale. Oop, and it looks like these didn't carry over too much, but these lines shouldn't be there. But I think we can work our way through this. So we had African populations, European populations, Asian population, and admixed population. On the y-axis here is just the number of derived alleles. So these are the number of variants that we identified in these individuals' different genomes. And these box plots just represent the average number of variants that populated those genomes. And as you'd expect, we saw a greater frequency of variants in individuals of African descent. And this is consistent with what we know about the founding of the human population, and where Africa is the most diverse population on the planet. Now, since there is a greater number of variants in the African individual's genome, there's a greater number of likely damaging variants in those genomes. Variants that are likely to be of functional significance. Greater number of variants, greater number of likely functional variants. However, if you look at the frequency of homozygous variants in the population, then despite the fact there's a greater number of variants in the African population, there's actually a greater number of homozygous variants in the non-African population. And you might ask yourself, why is that the case? Just has a little bit to do with migratory patterns and the effective population sizes of the non-African population. So after the migration out of Africa, a smaller subset of people than those living in Africa at the time made their way into the Middle East and ultimately up into Europe. That led to a smaller effective founding population size in those populations, which led to a higher kind of random inbreeding rate, which increased the levels of homozygosity. So again, this is nothing that I discovered. Many people know about this, but we've been able to catalog this on a genome-wide scale. So again, greater number of variants in African populations, but a greater number of homozygous variants in the non-African population. As a result of there being a greater number of homozygous variants in the non-African population, there's a greater number of likely damaging homozygous genotypes in the non-African population. In fact, if you just look at all the variant categories, functional variant categories, this is kind of consistent with that cartoon I had showed earlier. you can see that there is kind of a depletion in the non-African populations. These are negative coefficients, means there's less of a frequency per genome of these variants. Lesser frequency of likely functional variants in all categories in the non-African population. However, if you look at homozygous genotypes, there's a greater frequency in the non-African population. So this pattern is not confined to, say, coding variants or promoter variants, but variants of all types that might actually impact gene function. And these results are all highly statistically significant, and you encourage you to look at that paper if you're interested. Since the publication of that paper, there have been many other papers that have shown similar patterns, including those from groups working with the 1,000 genomes data and other data sets in the public domain. Now, what does this have to say about that filtering strategy? Well, it's pretty obvious. If your patient, say, is of European descent, but your reference population is made up of individuals of African descent, then you're likely going to claim that a variant in that European's genome is novel, when in fact it's not. It's just you're comparing it to the wrong reference population, right? So this is intuitive, given everything that I said. So these little plots here just show the number of false positive leads you might have in trying to determine what variants are novel in the genome of a patient with an idiopathic disease if you use the wrong reference population. And of course, we could turn this around and ask, well, what if our patient was of African descent and we only had a European panel to compare his or her genome to? So the moral of the story is you'd need a appropriately diverse reference panel. And again, this seems obvious, but in the context of this filtering strategy, nobody had really ever shown this. So, you know, not that having one or two additional variants that you might have to consider as a pathogenic would be a bad thing, but if in fact you've got hundreds of additional variants that may or may not be candidates for pathogenic, that creates problems. Okay. So it's one other thing that is very important for these filtering strategies is the attention that needs to be paid to phase information. So I don't know how you're familiar with phase, but when we sequence genomes with today's sequencing technologies, we don't get phase information by default. Now, what is phase information? Well, it's the nucleotide content of the two chromosomes, the one we inherit from mom and the one we inherit from dad. Typically, with sequencing technologies that are available today, you kind of get insights into whether or not a position in the genome is homozygous, non-reference, or heterozygous. But you're not actually given information about what variants were inherited together on a chromosome that came from mom or dad. In order to establish that, those kind of haplotypes, you'd need phase information. You don't get that by default. Now, why is phase information important, especially in the context of searches for variants that might explain idiopathic disease? Well, here's a little cartoon that I think could bring this out. So let's say there are two neighboring sites in an individual's genome where the individual is heterozygous. And say the bold nucleotides there are known to be damaging. They disrupt the functioning of the gene they sit within or around. Since this person is doubly heterozygous, we wouldn't know whether the following situation occurs, whether or not both of those damaging variants were inherited on the same gene homolog, such that we had one doubly mutant form of the gene and one wild-type form of the gene, or whether or not one of the damaging variants, say, occurred on the paternally derived version of the gene and the other one was on the maternally derived version of the gene. Now, why is this important? Well, it could be that the wild-type version of the gene can make up for the two mutations on the other one and lead to kind of normal function and be a compensation. Or if they're on opposite homologs, both copies of the genes are damaged, leading to a situation known as compound heterozygosity. And many rare diseases are known to be caused by compound heterozygous combinations of variants. So you need phase information. That might require you to, say, sequence the parents or leverage phasing approaches of other sorts in your strategy. So this is very important. And some time ago, my students and I had a review on the importance of phase information, often underlooked by contemporary genome sequences. So how did we apply this? Well, Lily Grossman, the young woman who had the idiopathic condition that I mentioned, whose story is written up in National Geographic and then recently in the Atlantic, had this idiopathic condition, had been confined to a wheelchair for the last 16 years. No one quite knows what's going on. Suffers from very severe nighttime tremors, so bad that it wakes up the parents in the middle of the night. They have to go running into the room. So a very brutal condition that she suffers from. So we sequenced her genome. This is two of my former postdocs, Ali Torquemani and Cinnamon Bloss, now both faculty members at Scripps and UCSD. And we subjected the variants to this filtering strategy, finding those that were novel from those that weren't, or likely novel, and then using the annotation tools that I had described to kind of narrow down the number of likely candidates for explaining the pathology. And we did identify a number of situations where Lily was a compound heterozygote, where the two non-reference variants were likely to damage the encoded protein. But there was one that made more biological sense than others, and that had to do with variants in this gene ADCY5. With some colleagues, we subjected these to functional assays, and it looked as though the ADCY5 mutation was a good candidate for explaining Lily's pathology. So on that basis, we then asked the question, well, what drugs were known to target ADCY5 and which might compensate for the problems induced by the particular mutation that we had? So we scoured the literature and identified a couple of studies that had looked at, say, knockout of the ADCY5 gene in mice that had then been hit with different drugs, et cetera, et cetera. And on that basis, settled on a particular drug that we thought might be of benefit to Lily with respect to kind of decreasing the number of severe nighttime tremors that she was showing. So we outfitted her with a device that could measure the number and intensity of the tremors that she was having at night, and then we gave her the drug to see if we could make a difference. And lo and behold, if this is the number of tremors she was suffering from, we initiated the drug, and they kind of went away. Now, sadly, this was initiated about 1.5 years ago, and since that time, she showed some resistance to the drug, so the tremors are coming back. But we had a short-lived success in identifying a drug that seemed to benefit her on the basis of identifying the pathogenic mutation and then going through the literature to see if we could identify a drug that might benefit her. Now, since the publication of her genome and those insights, an ADCY Commons, a research group has been started, and some 30 individuals worldwide have been identified that actually have ADCY5 mutation. So not only do we think we discovered the pathogenic variant, but we sort of kicked off a little campaign to find other people that suffer from this very debilitating disease who are likely to have ADCY5 mutations and who could benefit from an effort in repurposing drugs and identifying therapies and testing them out. Now, in conducting this study, I mean, we didn't need any statistics to really make the claim that the drug worked here, but oftentimes that's not the case. And we were lucky that the drug had the dramatic effect that it did in Lily's case. So the next question is, how could one design trials that might bring out effects in a more sophisticated way? So recently I had an editorial in Nature on the need for designing these trials, but this grew out of conversations that I've had for some time with the FDA about designing trials that are more efficient, either by leveraging genetic information or some way of selecting patients that are more likely to respond to the particular drug, and ultimately designing trials that really focus on the patient's well-being, not necessarily on the drug or intervention. And what I mean by that is many trials are done to make claims about the utility of the drug, and they're done on a population basis, right? You give, 5,000 people, your cholesterol-lowering agent, and 5,000 people, placebo, and then you see if the average reduction in cholesterol is greater in the active group than the placebo group. But what you're not doing is really asking the question whether or not any one particular patient benefited from that drug. You're making sweeping claims about the utility of the drug in the population at large. And that kind of flies in the face with this contemporary emphasis on personalized medicine. So hence the request from the Nature people for me. I'm very flattered to have been asked to do this, to put in a little piece on the need for individualized trials. So then how does one design these studies? Well, it's pretty simple in orientation, not so simple in implementation, and here's how it goes. Let's say we have some measure that we're interested in affecting via a particular drug, such as blood pressure. Well, we can measure a person's blood pressure. Then we'll give them a particular drug and see if their blood pressure drops, maybe take them off that drug, give them another one. Do this enough so that at the end of the day, we have enough data to make an objective claim about whether the drug is responsible for the reduction in that patient's blood pressure. And again, this is not what is often done in large-scale phase 3 trials, where you might get by with 1 or 2 measures on many thousands of people. So think of this as many thousands of measures on a small number of people rather than 1 or 2 measures on thousands of people, right? And of course, you could take another patient, pursue the same kind of trial in them, and maybe they responded to a different drug or whatnot. And I had a little review 4 or 5 years ago on this. Now, the design of these trials can leverage all the kind of statistical technology that goes into the design of large-scale phase 3 trials or traditional trials. So you can randomize the patient to which drugs they get at which time. You can blind the patient to what drug they're on. You can have washout periods. You can have placebo controls. They can be designed in a sequential or adaptive manner. You can look at multivariate phenotypes. There are some data analysis issues, probably the most pronounced that I'll talk a little bit about. This is not meant to be a P's and Q's talk, but I have a little bit of that. Serial correlation between the observations needs to be taken into account, as you can imagine. There might also be carryover effects and a whole slew of other issues that one would need to be sensitive to in carrying out these trials. And there are, of course, lots of extensions, which I'll touch on briefly. These studies have been pursued, and if you go through the literature, you'll see that many N-of-1 studies have been initiated and had IRB approval for and everything else that are documented in the literature. And I won't go through these, just suffice it to say that people have leveraged kind of single subject or N-of-1 trials with some varying degrees of success, looking at a whole slew of different conditions. Now, here's the most biostatistically oriented part of my talk. Talk a little bit about the design. You can ask questions. Well, how many measurements you need to be able to make a claim that a drug is actually working better than another drug? Do you need run-in periods, washout periods, et cetera, et cetera? So what I did with one of my former postdocs and a guy, Andrew Viterbi, and many of you may know Andrew Viterbi. He is the discoverer of the Viterbi algorithm. Every cell phone that people have got some signal-to-noise filter. It's probably based on the Viterbi algorithm. Andrew was a founder of Qualcomm, so he lives in San Diego. And I went to him and said, well, I think these N-of-1 studies with lots of data are kind of like signal processing things. And so, we sparked up a conversation and decided we'd work together. And some of what I'm about to say was the result of those conversations with him. So we have a simple linear model with autoregressive kind of serial correlation. Then we can look at kind of the power to detect signals as a function of the serial correlation strength, the effect size, the number of time points, et cetera, et cetera. So here's a reasonable question to ask. If one had a finite number of measures that they were going to make, how should you break up those measures throughout the trial? So let's say we had 2 drugs. Should you measure the person while on drug A and then stop and give them drug B? Or would it be better to give them drug A, then drug B, back to drug A, and then drug B? How would you break up the number of measures that you had with respect to what drugs they're given? Which would be more optimal? If there was strong serial correlation, would this design be better than that design? These are the sorts of questions that one could ask. Now, what about the use of washout periods? Would you strategically place them at certain points in the trial? How long would they be? Of course, that might be a function of, say, the half-life of the drug or other sorts of things. Or could you just let the number of observations collected while on each drug be kind of random? What is the optimal design? That was the question. I'm not going to go through this in tremendous detail. Just say we kind of worked this out mathematically, making the little assumptions about the AR process for the serial correlation. And what we found is you can make up for the strong serial correlation. This might be intuitive to many people in the room by breaking up the big treatment periods into smaller periods or collecting more measures, right? So if this is the strength of the serial correlation, and here is the number of periods, we could either have 2 very long periods or 40 very small periods where they're on the drug, just alternating back and forth between the drug, all with 400 observations made with a certain effect size picked up. You can see that the stronger the serial correlation could be offset by simply breaking up the number of time points. This may or may not be practical depending on the length of time you'd expect to see an effect on the drug, but it does give us some insight into how we can go about optimizing the design of these sorts of trials should we want to implement. Now, in order to carry these out, you need some way of measuring whatever it is that you want to measure, and unfortunately in today's kind of biomedical science community, there's a lot of emphasis on developing better phenotyping protocols. Talked with David earlier about this. I know he has a very big interest in this. So all sorts of wireless devices could be used. In the context of blood pressure, there's little Band-Aid-like devices you can slap over someone's heart that measure blood pressure, 24/7 heart rate. Got to worry about the reliability of these instruments, but suffice it to say that there's emerging technology that could be used to carry out the sorts of studies that I'm describing, right? Here is an example where we actually looked at blood pressure in a patient. We compared 2 drugs for participants with recently diagnosed kind of essential hypertension, so had never been treated before, but walked into the clinic and showed hypertension. And the question was, what drug would you give them? And this is often a question that plagues physicians treating people with essential hypertension for the first time. There's so many drugs a physician could choose from: beta blockers, ACE inhibitors, diuretics, you name it. So we did a little study on a patient and rotated the drugs, had washout periods, had kind of a funky design, but that had to do with the logistics of actually doing the study, because we did want to blind both the patient and the physician to the drug. So we had the pharmacy cook up pills that were identical in weight and color and everything else, including pills that were more or less placebos for the washout. And I can imagine this was complicated, but we were able to do this. We did it, and it looked like the patient's blood pressure went down to a greater degree while on drug B here, which was the ACE inhibitor. Unfortunately, when we measured the weight of the patient, or fortunately for the patient lost about 10 pounds while on the drug in this period. So when we treated weight as a covariate, we couldn't explain the greater drop in blood pressure to the drug's effects, but to her weight loss. So again, wonderful for the patient because she became more health conscious, lost weight, actually normalized her blood pressure. Not so good for the trial because we couldn't attribute the blood pressure drop to the drug. Another example of an N-of-1 study that we did had to do with addressing this issue of polypharmacy, something Brian and I have talked about with great detail. So we had a 60-year-old female who had been treated lifelong for anxiety, depression, and a genetic sleep phase disorder. So she actually had clock gene mutations that caused her to be up in the middle of the night and not sleep well. And as a result, she was treated with many different drugs, one for depression, one for anxiety, one to facilitate sleep. And all those interfere with each other, as is well known and documented in the literature, right? So the antidepressants impact sleep, but sleep aids impact mood, and hence it could exacerbate the depression, et cetera, et cetera. So it's kind of an ugly cycle for a patient to be in. So we designed a study where we test different combinations of antidepressant drugs used to maybe enhance sleep, et cetera, et cetera. And then we used a number of what were available at the time, devices to measure sleep, restless legs, activity, vital signs, and whatnot. I won't go through all the results. You can read about it in the publication, which was just put online at the journal F1000. But we did establish times in which we're going to try different dosages and different combinations of the drug, including some where she was on no drugs to kind of get a good baseline or what would amount to kind of a washout period. The upshot. Oh, I think so. I think, right. There are costs associated with conducting these trials. There's no question. But maybe with the availability of wireless devices and the amenability of patients to undergo this, it wouldn't be huge costs, and they probably offset all the downstream costs you paid for by having side effects and all the problems associated with not giving optimal treatment. So there's a real balance in terms of the economics there to be waged. Well, the bottom line of this study was, if you looked at, say, deep sleep, the amount of time the patient was actually sleeping restfully, the more drugs she was on, the worse. And the fewer drugs she's on, the better. So the take home was this, is that she actually did better when she was on fewer drugs, right? Which isn't going to make the drug manufacturers happy, but certainly made the patient happier. Now, one thing that did happen is when we removed the drugs, we were measuring vital signs. We uncovered sort of a sleep apnea condition. So her breathing changed when she was not on the drug. So she wasn't induced into kind of some sleep, and that unmasked this sleep apnea condition. So kind of created a situation where, geez, how are we going to go about optimally treating this patient if she's got all these exacerbating conditions, right? So this is a problem, and maybe the perfect setting for actually conducting these trials, because her condition was very nuanced. There's not everyone walking around who has exactly the same condition as this particular patient. But she was one who, for 40 years of her life, was trying to deal with all the clinical conditions that she in fact suffered from. Okay, now with these N of 1 trials, of course, you can conduct them on a single person, try it on someone else, find out if they responded or didn't respond, do this for a bunch of people. Then you've just brought out a phenotype in a very objective and sophisticated way. Once you've identified people that do and don't respond to drugs unequivocally using this data, then of course you could ask what they have in common, and it could be that they have certain genotypes in common or a certain omic profile. And in that way, in the future, on the basis of the genomic profiles, decide who should get one drug and not have to resort to the N of 1. So the N of 1 trials can also be used to actually bring out phenotypes that you otherwise wouldn't obtain if you didn't go through this more sophisticated and elaborate process. Now, there are some people, in fact, Susan Murphy at the University of Michigan, who's designing kind of optimal ways of conducting such trials. I'll just give props to her work, knowing that I'm at the University of Michigan, and say these are very sophisticated and wonderful approaches to conducting personalized trials. So I'll draw attention to her work. Okay, so you guys are all familiar. I don't see why you wouldn't be, but she is really pushing for more efficient and optimal trials to benefit patients, which is wonderful. Okay, so now bounce into kind of the third topic I want to talk about in cancer genomic trials, this concept of vetting algorithms versus vetting compounds. And again, if there are oncologists in the room, just bear with me if my first couple slides are really basic. So this table, which is more of an eye chart, and I apologize, can't really read it, just reflects some state of the thinking in patient genomic profiles, tumor profiles harvested from patients, and what drugs might be the best to throw at that cancer given what was found in the patient's tumor. So if in fact a woman has breast cancer and there's HER2 overexpression, then you should give Herceptin. If someone has EGFR overexpression in their colorectal tumor, then an EGFR inhibitor should be given. The community as a whole is developing insights into these rules that kind of match drugs to tumor perturbation, right? And they're growing in number. In fact, this little review paper by Richard Simon is somewhat dated, but the list of drugs and targets that have been identified is growing by leaps and bounds. The problem with this is if one wanted to actually vet the utility of giving that drug in that condition, you might have to conduct a large number of trials, testing each one to convince someone that that match is appropriate, right? And that's complicated by a number of factors. Now, my group over the years has tried to identify mutations that are likely to drive cancer and hence be targets, and we're not alone in this. Many, many other groups pursue this sort of thing, but we've published papers over the years describing how one might be able to predict which mutations identified in the tumor might be driving tumorigenesis and hence be the targets for specific therapies. And we've worked with different groups to show that these predictions actually do work out either in vitro or in the clinic. Now, how does one actually then test whether these rules lead to better outcomes than, say, the standard approaches to treating cancer. Well, this emerging trial design known as basket trials and umbrella trials have been proposed, and there's a simple intuition behind those. You just enroll patients, maybe with different tumor types, profile their tumors, find out what the mutations are that are present in each one of these, and on the basis of what mutations are present in their cancer, steer them to a basket that just reflects what drug they should be given. So for example, patients with mutation 2 here are steered towards basket 1. Patients with this particular mutation are steered towards basket 2. Now, what's important is you have this algorithm for deciding what basket the patients should go in. There's some strategy, some scheme that maps the drugs to the patient profiles and vice versa. And questions arise with respect to actually conducting these trials. For example, what is the algorithm based on? Intuitions, mouse models, clinical experience, what is it? What about the fact that there might be multiple genetic perturbations? In which basket do you put someone who has 2 likely driver mutations, right? What about combinations of treatment, right? These questions all arise in the context of basket trials that people conducting these trials are certainly thinking about. I'm claiming no originality for pointing out these issues with basket trials, right? And there are many trials like this going on. Many of you may know about this. I'll have a little bit to say about the Sheba trial because it basically showed that the people who were given the genomically guided therapies didn't have any greater benefit than the people who didn't. And there are reasons for that. Their algorithm was likely flawed because it turned out that only one of the baskets that they lumped people into is really filled with enough patients to be able to be powered enough to make claims about the utility of that match. So their algorithm was flawed, right? That's the point. But there's many other trials, including G.E.M.M, which is one that I'll talk about in a minute that I'm involved in. Now, this matching on the basis of the belief that a drug targets a specific driver mutation is also flawed for the reasons that I alluded to, that patients typically have more than one driver mutation present in their tumor. So it's not the case that genes work in these nice linear pathways where if there's one perturbation, we just need to correct it. Instead of genes work together in networks that are replete with feedback and redundancy mechanisms such that if you target one gene, another one compensates, blah, blah, blah. With Ali Torkamani, my former student and postdoc, we published papers that many other people have published papers showing this to be the case. There's a unfortunate but well-known story about a man with metastatic melanoma who was found to have a BRAF V600E mutation in his tumor, which suggested that he should get a drug called vemurafenib. That patient was given vemurafenib, and miraculously the tumors melted away. But because there was another mutation present in the cells that they're decombated, 8 weeks later, the tumors came back and the man died of the disease. So it's well known that if there are multiple drivers in a mutation, targeting one might not suffice, right? That's the problem. And in fact, Bert Vogelstein and colleagues over the years and others have published papers just kind of cataloging the number of likely driver mutations present in a single patient's tumor. And although it varies from cancer type to cancer type, you can see that there might be a large number of likely drivers of tumorigenesis present in the cancer, suggesting that simply targeting one of those with a drug might not suffice and work well. So this leads to then questions around how can one match drugs or combinations of drugs to the tumor profile of the patient. And there's multiple rules or algorithms one could exploit that the community is thinking about. One way is to just leverage multiple simple rules. So if someone's tumor has EGFR overexpression and HER2 overexpression, you give the EGFR inhibitor and Herceptin, something like that. I'm just making that up. You just use multiple rules. You could match drugs on the basis of how the collection of perturbations might be influencing a particular network of genes. These are all network medicine. You could just build de novo prediction models. Get a bunch of cell lines, look at expression patterns across the cell lines, hit those cell lines with different drugs, and then build predictive models, build regression models or whatever you want that might be used to predict what drug someone should get given their genomic profile at talk a little bit about that because I've been involved in some research doing this. The connectivity map is a tool out there to kind of match drugs to the multiple perturbations that might be present in a tumor or a cell line of one sort or another. I won't go through this in great detail, but it was designed initially by some people at the Broad. It's available in the public domain. If you have the profile, gene expression profile of a cancer, you can run that profile against the connectivity map, kind of query what drugs appear to reverse the perturbations in the tumor. So if in fact a gene was overexpressed in the tumor, underexpressed in the tumor, overexpressed, underexpressed, you'd want to find a drug that appears to counteract those and kind of reverse that process. And you could do that with the connectivity map. So it's a different approach for matching drug or a drug to a tumor profile. Or you could combine all these. Now, this is just a smattering of the different algorithms that people are thinking about in terms of matching drugs or combinations of drugs to tumor profiles. But there's other strategies or algorithms that are implicit in ways people treat cancers. And I'm not going to go through all these. I'll just give you one example. Emerging research into immunotherapies, where one could find kind of neoantigens in the patient's tumor. And on that basis, cook up a cocktail of T cells that have been sensitized against those neoantigens and then put them back in the patient's body so they could go and kind of kill off the cancer cells. Now, think of each patient as having a different neoantigen profile. So the cocktail of T cells that's cooked up would be different from patient to patient. So now what you're doing is not testing the same constructs against each patient. You're basically testing a strategy, finding the neoantigens, cooking up the T cells, putting them back in the person's body. So that's unlike testing a standard drug where you have the chemical composition of a particular drug and you're testing that on everyone. This is a strategy, right? And many other strategies exist, including whatever strategies a physician resorts to when they decide what drug to give somebody. Whatever the collective thoughts are, whether they go to Google and look online for papers that might sway them one way or another, that's a strategy of sorts. And it's not codified, but it is some kind of strategy. Something to think about. Now, people have proposed different algorithms and made claims about their ability to match drugs against tumor profiles. So a little less than a year ago, Patrick Soon-Chong, just 2 hours north of me in LA, made the bold claim that he has created the Google of genome mapping that is going to lead us to the nirvana of coordinated delivery care. So what he's basically saying, his Providence health system has algorithms for matching drugs to tumor profiles that will work better than whatever else the community is doing. And he's gone on record for saying this. Now, nobody's quite sure what is in the little black box that he has that matches the drugs to the tumor profile, right? This is considered a trade secret or proprietary. Not so long ago, a group of researchers argued that they could use Watson, the IBM computing system, to kind of match drugs to tumor profiles. So again, leveraging some kind of scheme or algorithm. So the question is, if these different approaches to matching drugs to tumor profiles exist, does it behoove the community to vet them and show that they actually have utility, much in the way you would test a particular compound to show that it has utility? Who's to say that the Soon-Chong algorithm is not better than the Watson algorithm or vice versa? Should the FDA take it upon itself to pit one of those algorithms against another to show that one has utility in the same way that you would pit Lipitor against simvastatin in a large phase 3 trial to compare the two? These are emerging questions. Well, I'm involved in a large study funded by the Stand Up to Cancer organization and the AACR to see if algorithms for matching drugs to tumor profiles obtained from patients with a unique melanoma condition, BRAF wild type melanoma, has any utility over the standard of care, whatever the standard of care is for people with BRAF wild type melanoma. A lot of researchers have been involved. This project has been running now for about 3 years. I'm not going to go through the results because they're not complete yet. Jeff Trent at TGen and Pat LaRusso, who's now at Yale, but was at Hermanos, just east of here in Detroit, is the clinical PI on the project. With my former postdoc, Ali, and Christina Vori at the Stanford Burnham Previs Medical Research Institute, we're involved in actually coming up with those algorithms, leveraging whatever we can from the public domain, like cell lines and whatnot. Hence my sensitivity to these issues. It took about 2 years to get this protocol through the FDA for reasons that will become clear in a minute. Here's just a few factoids about the trial. We actually have 37 drugs we can choose from. And 17 of those are investigational agents. So drugs that passed phase 1 but aren't widely distributed that the pharma companies gave us to act on. So once we profile a patient's tumor, we look at the perturbations, then we can choose among these 37 drugs to give to the patient, right? But we have to come up with some kind of algorithm. And I'll talk a little bit about that in a minute. Suffice it to say, when we started the trial, we were told we had to lock down the rules for matching the drugs to tumor profiles. And this is highly problematic for reasons that will become clear. Yeah, I did. It was an absolute nightmare. Now, people have proposed that there's other pieces of information one could leverage to decide what drugs to give a patient. So you could take someone's tumor, make a cell line out of it, hit that cell line with all sorts of drugs, and on the basis to how the cell line responds, give the drug to the patient, right? This has been done, a recent paper by Engelman's group in Science not so long ago suggested it. But there's a problem with looking at cell lines and drug responsiveness for kind of guiding patient care. And that has to do with the fact that many people believe they're unreliable. And in fact, a couple of years ago, John Quackenbush and colleagues published in Nature a paper questioning the reliability of drug screens against tumor cell lines. So it's got a lot of attention. It was in, probably the best journal on the planet, Nature, and it caused a lot of concern. Late last year, the group that was responsible for conducting these screens in the CCLE and the CGP, responded, again, published in Nature, saying that no, the results of their screens were reliable and reproducible. Well, it's a little bit funny, and I encourage you to read the paper, so take what I say with a grain of salt, is both produced kind of correlations, reproducibility, somewhere between 0.4 and 0.6. So really it came down to interpretation, whether that was considered evidence for a strong reproducibility or whether it was not strong enough to be considered reproducible. So go back and look at the data. It has more to do with interpretation and a few differences in analysis. Very interesting. So we did in the context of the Stand Up to Cancer, knowing that there was this controversy over guiding patient therapies on the basis of cell lines, is we did an experiment. We sent out cell lines to 2 different labs. We used 9 different doses of hundreds of drugs, and we just treated it as a big analysis of variance. So we had as factors the laboratory in which the experiment was done because they were reproduced. We had the drugs. We had the dose of the drug. We had the plates on which the different cell lines were set up. So we could actually look for plate effects. So just partition the variation in the entire experiment into different sources and ask the degree to which those sources contributed to this. This was not done by those 2 other groups, by the way. They just used kind of IC50 values abstracted from dose response curves. But we went back to the experiment as a whole. What we found, maybe not surprisingly, we didn't see a whole lot of lab-to-lab variation, although they did. We did see pronounced drug and dose effects, as you would expect, and actually some, importantly, dose by drug effects, suggesting that some drugs really had a more pronounced effect in cell lines. We also saw that some cell lines responded very differently to others. Keep in mind, this is in the context of the entire experiment. So it wasn't any one drug we were looking at, getting IC50s and then looking at things. It was across all the experiments. How long? That's a good question. So I think you want to talk to Christina and my colleagues at TGen. They just gave me the data. So what the ultimate experimental conditions were, you'd want to talk to them. But cell viability was the outcome that we had looked at. So these questions are important. Sure. Sure. No, you're absolutely right. There's a lot of nuance to this. I mean, you could do co-culture experiments. You could do all sorts of other stuff to guard against safeguards, whether the drug actually had an effect on the normal tissue versus, I mean, there's tons of stuff that you could build in. And I'm not an expert on the optimal way of designing the wet lab side of this. I just wanted the data to see if we could make some broader sense of it. Now, we did actually see plate effects. So maybe what wasn't reproducible in the previous studies was a couple of plates showed outlying values. And if you got rid of those and then went back and reanalyzed the data, you'd see greater consistency. So we did see some of this phenomenon. Now, in analyzing the cell lines, this is, again, more anecdotal than anything else. You know, we had the cell line. It was hit with a bunch of different drugs. We saw some that didn't do much of anything, some that appeared to have a pronounced effect. In modeling, this is more for the biostatistics people in the room. In modeling this, again, our approach was not to kind of extract IC50 values and then use those as summary measures and then compare them. We went back to the data using more of this analysis of variance-like approach. So what one could do is if you had a bunch of cell lines, say 30 of them, you could extract IC50 values and then look at them. Or you could go back to the raw data and consider, you know, all the information that you had at your disposal to decide what drugs were and weren't having an effect in the context of the whole experiment. So that's something we're working on. Now, in context of matching the cell lines to the patients, we did go through that exercise. These are 27 of the 30 patients gone through the Stand Up to Cancer trial, numbered here. We then have kind of color-coded different cell lines that look like they match the patient profile, and then we could read off, oops, sorry about that, read off what drugs we showed an effect in the cell lines. Now, these weren't actually tested in the patients, but we at least had this information to see if there was any likelihood that these drugs would have worked in the patients had they been given. Now, what one would like to have at his or her disposal is a database that has all this information at their fingertips. So if you had patient profiles, you could match them against a database that had cell lines that were also genomically profiled that were subjected to the different drugs. Tumor graft data could be used. You could implant a tumor from a patient into a mouse, hit that mouse with different drugs, look at responsiveness. This is also being done by many groups. Dogs are one of the few species that develop spontaneous tumors like humans. There's a lot of research in treating canine tumors with different drugs. You can build up data there. But of course, what you'd really want is actual patient profiles and their outcomes to match to others. And there are databases that are growing. Now, one problem with vetting the algorithm that we had in the Stand Up to Cancer that we discussed with the FDA had to do with the nature of our algorithm and what went into the algorithm. So we had to biopsy the patient, do the genomic profiling, right? Come up with the matching algorithm, give the results to a tumor board that then decided what drugs to give to the patient. And that's the way it did. So you might think, well, here's where all the action is, this matching algorithm. Well, they called into question the reliability with which we could do the sequencing, kind of garbage in, garbage out problem here. If we can't reliably assess the expression levels of the genes or the mutation profiles, then how could we make any claims about the existence of driver mutations that might be targeted with drugs? So they asked the question, well, is part of our algorithm, matching algorithm, not just the scheme for matching drugs to the tumor profiles, but actually identifying the mutation profiles themselves? The other thing, there's deliberation by the board of experts. The tumor board looks at our results and on that basis decides what drug to give the patient. Should those deliberations be considered part of our algorithm or strategy? And of course, that would definitely vary from hospital system to hospital system, right? Now, the other thing that was a little bit complicated in this is the people that we were studying, the people with BRAF wild-type melanoma, typically have, when they're diagnosed and been on all sorts of other drugs, 3 to 4 months to live. So we didn't have the luxury of time in making these decisions. So we had to do all the profiling and the matching of the drugs in less than a week. And that is a problem, as you know. But without that, it wouldn't do anyone any good if we're really trying to test this in the patient, right? Now, there's another fundamental problem with these designs that's come up that you may or may not anticipate. In the context of this trial, what was originally thought is we'd come in, get a patient, and randomize to getting the genomically guided therapy versus the standard of care. And then on that basis, provide them the drug. Well, this was thought not to be effective because patients coming in for which there wasn't a good match, like it wasn't obvious what drugs to give them on the basis of their genomic profile, if they were randomized to the genomically guided group, they'd water down the signal because there'd be no confidence that the drugs would actually work against them. So to safeguard against that, what we were recommended to do was bring in a patient, right? We then do the genomic profiling. If there are good matches for that patient based on the genomic profile and our algorithm for matching the drugs, then they're enrolled in the trial. If there's not, they're going to get standard of care anyway, so they're not enrolled in the trial. But if, in fact, they're enrolled in the trial, then we randomize them to getting the genomically guided therapy or the standard of care. Now, why is this problematic? Because many of the oncologists in the community, when they heard about this design, were confused and hurt by it because they said, well, if I have some intuition about a drug that's likely to benefit my patient, I don't want them the possibility of them randomized to standard of care. So we pitched that to the FDA, and they said, well, you're begging the question about whether your algorithms have any utility at all. You have to prove it first. So I have to say I'm on the fence about this one, but you can imagine how this created controversy. The trial was approved, and we're moving through with this design. With the patient. Right. They consent. So all this is consent. Now, in discussing this trial recently with the FDA, so a little less than a year ago, a number of issues came up when we talked about them. I'm not going to go through them all, just two that I will focus on. What if insights external to the trial data come up that might impact the physician's choice or the researcher's choice for drugs? What I mean by that is we had an algorithm for matching the drug to the patient profile. We locked it in at the start of the trial. What if the day after we started the trial, there was a Nature paper published, cover article that said, if this mutation is present, use this drug because we've shown in mouse models and in patient studies. If we're locked in and can't include that information, then we wouldn't not only benefit the patients, but we might be shooting ourselves in the foot with respect to testing a useful algorithm. So the compromise was that we could have periodic changes in our algorithm over time to accommodate this sort of thing. And this has actually impacted oncology trials. So there was the BATTLE trial, lung cancer trial that Scott Littman, the director of the UCSD Moores Cancer Center, was involved in, where all this information about EGFR inhibitors came out after the trial was initiated. But they were locked into the little buckets that they guided people to and couldn't incorporate that information, right? So that's a problem. So one has to be flexible in the way they conduct these trials. Now, this raises important biostatistical questions for interpreting the trial results. Because if you're allowed to adapt the rules that you use, then the patients enrolled at the tail end of the trial are going to have access to a better algorithm than the people early in the trial. So then you have to weight the patients differently. This gets complicated as well. But this is a reality. Okay. Now, in conducting these trials, you need the appropriate infrastructure. And here I've had the pleasure of working with a group involved in the MED-C consortium. I don't know how many people know MED-C. But as we talked about before, if you wanted to test all the different matching schemes that one could come up with, how could you do that? What infrastructure could you leverage? It would cost the pharmaceutical industry and NIH zillions of dollars to pursue all these little trials. So what was proposed by a number of people, and Mike Doherty from Roche Genentech is one of the leaders in this, was to put together a pre-competitive infrastructure for carrying out trials in oncology that would benefit all pharmaceutical companies. Okay? So the idea here is that if each pharma company ponied up some tens of millions of dollars to build infrastructure where they could recruit patients, then each one could benefit from having a central group recruit patients, and then they conduct trials with those patients, steering them to the right baskets based on their genomic profile, right? If they didn't do this, then each pharma company would have to go out and reproduce all this infrastructure, and it cost them billions of dollars. Okay. Oh, am I? All right. So I will just whiz through this. What is a personal versus population threshold? Well, if you're measuring the level of a biomarker and you want to decide whether someone has any signs of disease or not, You'd do this on the basis of some population threshold. So if this is cholesterol level, this would be greater than 200, right? But if you had established kind of a historical precedent for a patient, you might want to rely on that historical precedent to determine what is or isn't a reasonable value for that patient to have in the future. So for example, if this is cholesterol level and this patient had low cholesterol levels well below this population threshold, and then you saw in subsequent values they had elevation, then despite the fact they haven't reached the population threshold, on the basis of their deviating from previously established thresholds, you then claim that something is going on, right? So this is a personal threshold versus a population threshold. And I'll whiz through this and suffice it to say that there's a recent paper showing the utility of personal versus population thresholds and looking at CA-125 levels in ovarian cancer. And what they found is if they had used the personal threshold to determine who had signs of ovarian cancer or who didn't, rather than the population threshold, they could have captured the ovarian cancer almost a year earlier. So I have a study studying people that are highly susceptible to cancer, where early detection is absolutely crucial. So these are people with germline p53 mutations, other sorts of inherited cancer syndromes, where early detection is important. You go up and look at this. There's many other conditions that have this. So what we've done is actually established a protocol to measure frequently many different biomarkers, establish personal thresholds, and on that basis see if there's any signs of cancer. So again, these are people that almost have a 100% chance of developing multiple cancers throughout their lifetime. So early detection is absolutely crucial. And we've been pushing this. There's different signals that we've tried to detect from the data, multivariate signals. Not going to go through that in great detail. We had to design studies to deal with assay drift over time. Most longitudinal studies, you'll get all your samples and you'll analyze them together in one fell swoop at the end of the study. If you're doing things in real time, you don't have that luxury. So you have to do the assay, then get another sample, do the assay again, and that assay-to-assay variability, especially with metabolomics, where there might be changes in ionization patterns, that can come back to haunt you. So you have to design ways of minimizing these technical artifacts. Okay, the last point I wanted to make had to do with a big study that I'm involved in, looking at factors influencing lifespan. We've got two different communities where we're looking at this, one in Białystok, Poland, one in Cilento, Italy, very contrasting communities. We've decided to sample the individuals for this study through households. So we're going to get, by default, people that are related, right? We'll have individuals, we'll know their pedigree links, we'll then have households. So we can use kind of variance component models. Ooh, that doesn't look good. It must have switched over wrong. We can use variance component models, which I'm not going to go through in any great detail. This didn't show up in the transfer from my PC to my Mac. But we can partition the different sources of variation, right? And ask how different factors contribute to these health-related parameters. And the reason I wanted to point this out is 20-some years ago, I worked on this as part of my PhD thesis. It turns out that two of my thesis advisors also worked on this. This was an inspiration to me. That's what led me to think about these models. And of course, someone else in the room also participated in this. My father published papers almost 40 years ago suggesting that one could actually leverage family-based designs to look at household effects versus genetic effects and break down that variation. So the moral of the story here is you can take the researcher out of Michigan, but you can't take Michigan out of the researcher. So go blue here, and I'll thank my lab members. And if there's, I know I went over, I apologize. So thanks for listening. Pat, where's the hook?
So one thing you of course showed is that more information is better, right? This applies to, I mean, having so more information is better. That applies to knowing what the paternal and maternal chromosomes look like. Doing a longitudinal study and all this. How important is transcriptomics, for example, to looking at the RNA transcripts? How important is epigenetic marks, right? And how complete of an information set do we need?
Well, I think, you know, taking this more integrated approach is the way to go. And in the context of cancer, just to give you an example of the importance, you know, identifying the likely driver mutations in cancer just based on genomic sequence is likely to fail, because oftentimes you want to see that the mutations are in fact influencing the expression levels of the gene or the levels of protein of the gene, and you can't do that with just genomic sequence. So many algorithms to decide what variants are likely driving cancer from what not make the assumption that the mutations are activating. They might not be, but the way to know it is to actually interrogate the expression levels of the gene. So there's a case where the more integrated approach, I think, is essential for really nailing down what is and isn't driving the tumor. But in other contexts, it's equally as important.
In the case of cancer, one of the things we grapple with is tumor evolution. The tumor is evolving in the host, and a lot of what you're talking about, of course, sort of in a global sense, perhaps, samples the host factors, but not yet so specifically, because we don't always know what host factors. The CA125 is a nice example of that. It's probably unique. So the questions are to you is, there is the clonal heterogeneity, and that the tumor is evolving, and so even knowing we are capable at times of serial sampling, but not often. So what are your thoughts about longitudinality in cancer monitoring?
So yeah, obviously I didn't have time to go over a lot of this, but yeah, people are thinking about liquid biopsies now to kind of trace the evolution of the tumor, either you know, it's natural history prior to treatment or after treatment, and so those could provide insights. One thing that I would emphasize that is consistent with part of my talk where I talked about algorithms is, if you shape therapy on the basis of what you see over time, then again, you've got like a scheme or an algorithm. So if you say, okay, I'm going to start out giving this drug, and then if over time I see, say, from the liquid biopsy or in a repeated biopsy of the tumor, a different set of mutations arise, then I'm going to switch to this treatment. Then you might ask yourself, should you have to, you know, what is that? On what basis are you making that decision? So there could be a temporal component to the scheme or algorithm that's being implemented. It just adds another dimension to things. But then you'd ask yourself, well, does that have any merit? And the question is, would you have to test that? So it's just like a series of conditional statements. If this is seen at time 1, but this is seen at time 2, you kick in, you know, different therapies. It's just a different strategy. And much of this is simply, you know, the practice of medicine, physicians making decisions on the basis of what they see, and maybe they would or wouldn't benefit from kind of objective claims about what might and might not be called for clinically. But just because you have this temporal component doesn't mean it's not part of some broader scheme or formal way of deciding what drug to give a particular patient.
Would you say a bit more about statistical analysis in the G.E.M.M trial? Sounds as though you don't have time to do a crossover trial on each patient.
Right. No, we absolutely cannot. Not only do the patients not have enough time, but ethically, just putting them on a drug, you know, that they might not benefit purposefully to show that the one drug be better raises ethical concerns. That's probably not the case for, you know, treating hypertension. So there are some conditions that, you know, you wouldn't want to throw at the N of 1 design that I talked about with blood pressure in this context. You wouldn't want to do that. But statistically, what the FDA decided on, my father was involved in some of these discussions, was a simple design. It's the PFS, the progression-free survival, and it's the experience of the people with the genomically guided versus the standard of care. That's it. It's like a 2-by-2 table. On that basis, we're going to make the claim whether the genomically guided therapy works better or not. That's what was settled on. However, there's all sorts of post hoc analyses which we could do. If the algorithm shifted throughout the course of the trial, then this weighting the patients could be done, et cetera, et cetera. But the primary analysis is so simple in orientation, you know, to really defy the complexity and the sophistication of everything that goes into it. It was just, what is the experience of the patients in the genomically guided versus the standard of care group?
So exactly on this, Mark, one of the issues why people prefer having these double-blind, placebo-controlled trials is, and not a trial of one, is the placebo effect. And I could imagine that a woman, and in fact, I'm kind of doing a trial right now, and I'm afraid of the placebo effect because the people know that, you know, oh, some big researcher somewhere in America has done something that suggests they should try this drug. I mean, how do you know that when your drug has an effect in this one person and it deviates from the standard, from the number of, you know, seizures at night or whatever, isn't because they know that they are in such a wonderful new algorithm, etc. environment? How do you do placebo control in a N of 1 trial?
Well, we could have given, say, the patient in the antihypertensive trial a placebo and blinded them to that, so they wouldn't know, right? So it is possible to do those. There are ethical reasons why that approach wouldn't work in other contexts, like the cancer case. You wouldn't knowingly take someone off a potential effective med. In the blood pressure example, you might do that because it's known that the placebo effect can be so pronounced. You know, in that context, I'll just share with you, I have contracts from Janssen to kind of look at the data from some of their large phase 3 trials. And of course, it's in the two worst areas, I think, for placebo effects, looking at their experience in antidepressants and in pain medicines. These are the, right. So I said, you gave me the worst of all situations to work with respect to placebo. So it's crucial. Yeah.
All right, why don't we think next step?
Yeah, too, and the polypharmacy stuff.
Yeah, polypharmacy. We can talk about it.
Yeah.
Because I told him to.
What's this, the placebo effect?